At ASH this year, we are presenting the interim analysis of the ZUMA-12 study, which is a phase 2 multicenter study evaluating Axi-Cel as part of first-line therapy for patients with high-risk large B-cell lymphoma. Axi-Cel was previously approved for relapsed refractory large B-cell lymphoma after 2 or more lines of therapy. In this study, we evaluated Axi-Cel as part of first-line therapy in patients with high-risk large B-cell lymphoma, which is defined as patients with the diagnosis of double hit or triple hit lymphoma or large B-cell lymphoma with IPI score of 3 or greater. In addition, they had to have a dynamic risk assessment as well after 2 cycles of standard frontline chemotherapy such as R-CHOP or R-EPOCH. A PET scan should be FDG positive with a dual score of 4 or 5 to be eligible for the study. We enrolled a total of 32 patients as of the data cutoff who received cyclophosphamide and fludarabine conditioning chemotherapy, followed by a single infusion of Axi-Cel at a dose of 2 million CAR-positive cells per kilogram body weight. The median follow-up on the study was a little over 9 months, and this was a planned interim analysis. The best overall response rate on the study was 85%, and the complete response rate was 74%. And as of the data cutoff, 70% of the responses are ongoing. The therapy itself was quite well tolerated. Almost all the patients developed cytokine release syndrome, but these are mostly grade 1 and grade 2, with 9% of the patients having grade 3 or higher CRS. Neurological toxicity of grade 3 or higher was observed in 25% of the patients, but these were all reversible. There were no grade 5 adverse events due to CRS or neurological toxicity. We also found that the Axi-Cel product had a higher frequency of naive and younger phenotype of CAR T cells that were CCR7 positive and CD45RA positive compared to new Axi-Cel products used in the ZUMA-1 study evaluating this product in third line and beyond. This was associated with a greater expansion of the CAR T cells in these patients compared to the ZUMA-1 study. So in summary, in the ZUMA-12 study where we evaluate Axi-Cel as part of first-line therapy, Axi-Cel was found to be safe and it demonstrated substantial clinical benefit in patients with an unmet medical need. The high overall response rate of 85% and CR rate was 74%, with 70% of the patients had ongoing response after a median follow-up of 9.3 months. And the higher frequency of the naive phenotype of the CAR T cells in the pre-infusion product was associated with greater expansion of CAR T cells, suggesting improved T cell fitness in first-line treatment. So this is the first study evaluating CAR T-cell therapy in first-line therapy. While this is an interim analysis and further follow-up is necessary, these early results indicate that CAR T-cell therapy can be highly effective even in very high-risk large B-cell lymphoma. If long-term durability of these responses are confirmed and this efficacy could be demonstrated in a much more larger study, this could potentially change how we manage patients with large B-cell lymphoma at initial diagnosis in the future.